Hi again, everyone. Welcome to our discussion on Chapter 3 in Bio 165, the chapter about water. So here's a question for you that I'm going to talk about over the next couple of slides. Why is water so important? So important that that the Romans actually invented a word, rivalis, meaning using the same river as another. Rivalry is where it comes from, the word rivalis. Whiskey is for drinking, water is for fighting over. Mark Twain. And then statement after statement from national and international leaders about the fact that we might be at risk of wars over water. A few years back, about when I was just out of college, we almost got into a shooting war with Mexico over water from the Colorado River because the United States was literally using every drop of water, and none of that water was actually making it to Mexico. So if you look at a map of Mexico and you see Baja California versus the main area, that little inlet between Baja California, that's where the Colorado River is supposed to go. But hardly any water makes it there because it's just so important. But the importance of water cannot be overstated. All of these conflicts have been going on for decades or even millennia. The basis of the Arab-Israeli conflict is over water sources. In the Six-Day War and in the wars in the late 60s and 50s, what was happening was the Arab forces came in and were cutting off water to the main part of Israel. All of the water supplies for Israel come out of the Golan Heights in Syria, West Bank, which is traditionally now Palestinian area. So before that, the origins of the Arab-Israeli conflict are not rooted in religion. They're actually rooted in water supplies. In Bolivia, not too long ago, there was an effort to privatize water sources, and the city rose up to the point where the protests got so violent they were bringing in troops over water. The border wall, the caravans that you've been hearing about in the news, and this frenzy over building the border wall, that is due in part to the fact that there's been a drought in Honduras and in the southern Yucatan Peninsula for a few years, and the people just don't have any, they are starving to death. They are dying of drought. And so they are trying to come to the United States, not because they are seeking some sort of easy life. It's because they can't continue to live in their area because of lack of water. Even the Flint water plant, that has been a major crisis. It's back to water. Again, why is it so important? A border dispute in Tennessee and Alabama. Georgia is trying to get the border of Georgia, Tennessee, and North Carolina shifted about 20 or 30 miles north. And you know why? It's not because they want the land, it's because they want access to the water. And you can see conflicts, again, lasting millennia. And here's the really troubling part, is that this is just in the United States alone. These are conflicts over water in the United States. Look at the trend. It's exploding. It started exploding. Now, a lot of that may be due to economic development, but not really. I mean, we've been having a lot of economic development since the 70s and 80s. This onset is almost directly climate related. So again, I ask you, why is water so unbelievably important? And the answer to that lies in the information I'm about to give you over the next 19 slides. Water is essential for life, and it has chemical characteristics unlike any other compound that we know. Now, if we send a probe somewhere in the solar system and we find what we think is life that doesn't associate with water, that would literally change everything. But so far, what we know is that life depends on water, and that has actually driven our exploration efforts, is that we start looking for water in the solar system, and that's where we think we might find life. Water is polar. It is a polar molecule, which gives it interesting solute properties. The water has this polarity to it, this partial positive and partial negative because of the difference in electronegativity. That's why this is pink. This is showing you the likelihood to find an electron in this area versus this area. This polarity gives it unique solubility properties and so forth that we're going to talk about. And that magic of water is due to the hydrogen bonds that it forms with other water molecules or other compounds. This partial negative interfacing with a partial positive on another water molecule. That hydrogen bonding is going to give rise to what we call emergent properties that are essential for life. And here's just another stick figure. These solid lines, covalent bonds, these dashed lines, hydrogen bonds. Remember that hydrogen bonds form between a hydrogen and either an oxygen, like in this case, or a nitrogen delta negative in the case of things like DNA. But you can spot a hydrogen bond a mile away because of the dotted or sometimes dashed lines. And so we've identified 5 emergent properties about water that make it so utterly indispensable for life, indispensable for the ability to survive and grow and reproduce. Certainly that hydrogen bonding is at the core of it. You wouldn't have necessarily predicted, if you look at an O and 2 H's, that it would form such extensive hydrogen bonding networks with other water molecules. Cohesive and adhesive behavior. And I'll give you some examples of that a little bit later, but I want to set you up here. Why is it, let's say for a moment that we put you in a special shower, but instead of water, it drops pea gravel. The pea gravel hits you and it bounces right off and it doesn't stick to you at all, or not normally. But when you get out of the shower, the water clings to you. That's a little bit about cohesion and adhesion, and that has to do with hydrogen bonding. The ability to moderate temperature. If you take physics or chemistry, you'll often talk about specific heat, and water has a high specific heat. It's able to absorb and in some cases slowly release heat in order to keep temperature relatively stable. The expansion upon freezing, the fact that that ice crystals have a less density than liquid water, is a critical thing for especially in the case of Earth, but probably on other planets as well. Versatility as a solvent. If a molecule has any semblance of polarity in it, there's a pretty good chance that water will dissolve it. So those are the 5 emergent properties, and you should get to know those pretty well. Okay, because again, that hydrogen bonding drives these other 4, but hydrogen bonding is considered one of the emergent properties. Since we've already talked about hydrogen bonds, I'm not going to repeat those slides. I'm going to move on to the second emergent property, which is cohesion and adhesion. You can see that if you get it right, you can actually float a paper clip on the surface of water. Cohesion and adhesion. Cohesion is the property of water molecules adhering to each other with that hydrogen bonding. You can also use hydrogen bonding to other types of compounds like the wall of this vessel or this glass. We actually have a penny floating on the surface if you get it right, or water skaters, if you see those in the summertime, they're doing the same thing. They're taking advantage of the surface tension, and surface tension is created by cohesion. But there's also adhesion. Adhesion is the ability of water to hydrogen bond to other polar molecules, and adhesion is the reason that trees can transport water up sometimes hundreds of feet from the groundwater sources, because not only is the water sticking to itself, but it is also adhesion to the walls of the vessels in the trees. Adhesion is water forming hydrogen bonds with other molecules. A great example of adhesion is when you jump into the water or you get in the shower and you notice how your hair changes. That's because water is adhering to the hair, causing a slight change in the keratin structure. Cohesion and adhesion. And here's an example from your book showing about water and specific heat. Beyond the fact that you can control temperatures and make areas more amenable to life using the temperature moderation aspects for geography, there's also an important homeostatic mechanism that is known as evaporative cooling, and we see that as sweating. Or if you see a dog panting or a cat panting, that's because they don't necessarily sweat on their bodies, but they do sweat on their tongues, and so they use evaporative cooling to help control their body temperatures, and we use sweating to do that as well. That's because when we change from liquid to gas, there's a huge energy jump. And that's why, in fact, steam burns are often more severe than, say, a burn when you come in contact with a hot solid surface. That's because there's so much energy stored up in that gas that when it turns back into liquid, it releases all that heat. But that makes water a wonderful energy sink. If we evaporate liquid to gas, it takes a lot of energy with it. And so that makes it a very, very effective way of removing heat from a body system. That heat of vaporization is what it's called. It's how much energy, how much heat that a gram of water must absorb in order to shift from a liquid to a gas. Okay, so we call that evaporative cooling. And just like the land masses, it helps stabilize body temperature. It keeps it in what we call a homeostatic window. If we overheat, we'll have issues. If we get too cold, in some cases, we'll certainly have issues. Another major emergent property that makes water critical for life is its properties having to do with the density. As water, let's say we have room temperature water, and as we chill that water, the water is going to get more and more dense until it hits about 42 degrees Fahrenheit. At 42 degrees Fahrenheit, down to the freezing point, the water begins to get less and less dense, to the point that when it actually freezes, it's about 10% less dense than the liquid water around it. And that's why ice floats. That's why ice floats. If ice didn't float, if ice was more dense than water, that ice would sink, the water above it would serve as an insulator, and we'd make more and more ice, and the world would freeze over. That's cool. Pretty cool that it doesn't do that. Thank you for not doing that. There's another sort of more pragmatic aspect to it. If any of you like to do the deep type of fishing, like for instance to go up to Lake Michigan, or you like to fish in the big lakes or out on the ocean, turns out that density property is really, really important for fish. Fish will collect on these thermoclines because that's where food will collect because of the density change. It's really cool. So if you find where that water temperature shifts either dramatically or you hit that 42-degree mark, the food will literally collect on that density interface, and that's where fish will show up. Just a little bit of pragmatism. Because of that hydrogen bonding and polarity, that wonderful emergent property of water is that it can dissolve polar substances with relative ease. And so we have some terms here for you. The idea that we have a solution, that is, we have a homogeneous mixture of substances. So you have a solvent, in the case here is water. If we talk about an aqueous solvent, it means a polar solvent. And then we have whatever is dissolved in that solvent, we call solute. Usually if we have a polar substance, it will dissolve into separate molecules, like in the case of glucose, or it may dissociate into ions. So a solvent is a dissolving agent, and a solute is the substance that is converted from its original form into a homogeneous mixture in that solvent. And so we're talking about aqueous solutions in biology. The magic here is that it can dissolve them, but it also creates this equilibrium desire, shall we say. The desire to go from A plus B to AB, or back from AB to A plus B that I talked about earlier. So water can dissolve polar substances. So what we often see is in the case of salts, where we have a cation and an anion, you see those cations and anions dissociating. So here's a crystal of a given salt, and here is that salt that has now been dissolved in water. Now, larger molecules like glucose and so forth will form something rather than dissociating like this, because glucose can't break up. It's got those covalent bonds in it, that ring structure. It forms what's called a hydration shell. That is to say, there's a layer around the molecule that allows it to interface with water in a dissolved way. Those larger molecules, things that we call non-ionic polar molecules, that is to say they are not able to dissociate into ions. Things like proteins, soluble proteins, and a great example of this is albumin, like the egg white in an egg. They are polar enough that when they encounter water, water will form that hydration shell that I mentioned in the previous slide, and this will behave just like an ion. It will dissociate from the other proteins and form a homogeneous mixture that we call a solution. And some more terms for you. So if we have a hydrophilic substance, that is a substance usually because of its polarity or charge or ability to dissociate has an affinity for dissolving in water. A hydrophobic substance is one that does not have an affinity for water. It is afraid, like we talk about a phobia. Hydrophobic means that it does not have an affinity for water. On the counter side, we can talk about lipophilic and lipophobic. We don't usually use lipophobic all that much. We certainly use hydrophobic. A hydrophobic molecule will often be lipophilic. That is to say, it'll dissolve in an oily substance, a nonpolar solute, as opposed to a polar one. So hydrophobic is the same as lipophilic, and hydrophilic is the same as lipophobic. Just like oil and vinegar here. The black substance is a type of balsamic vinegar. It's hydrophilic, and then we have olive oil around it, very delicious. And you can mix it up and you can form what's called a colloid. It's a suspension of fine particles in a liquid or gas. In this case, it's a liquid. So you can break up these molecules and you can form this sort of suspension. They are not dissolved in each other, or they're very poorly dissolved in each other, but they will actually reform back into their original particles. Just like fog. Fog is actually a colloid. That's not steam. Those are water droplets. And that's why we typically see fog in cooler conditions, because it's not steam that's going on. It's water droplets. And eventually that water will settle out like in the form of dew. Again, cohesion and adhesion. So this next slide is going to be a tricky one. I want you to pay really close attention to what's going on here, because this sets the stage for a huge, huge concept that has life or death importance. Water, when it forms those hydrogen bonds with other water molecules, doesn't stay like this. What will happen is that every once in a rare while, in pure water, this is pure water, nothing else going on, one of these molecules will break down and you will form what's called a hydronium ion. It's actually an H3O plus. We usually just abbreviate it as H plus. Okay, and that's also an ion that can occur. But what happens is when this shows up in water, it immediately forms an association with a water molecule and forms this hydronium ion. You've probably heard about that in chemistry. But every once in a while, one of these molecules will lose a hydrogen to another one, and it'll form an OH negative and an H3O positive. About 1 in 10 million, okay? But when it does that, it will sort of cycle back and forth. So on average, we see for every 10 million molecules, we'll see one of these. If you can get that, then you can begin to understand pH better and why it is so critically important. So this is like AB and this is A plus B, or the other way around. In any case, The more we have of this, the higher that frequency. If we get above 1 in 10 million, like say we go to 10 in 10 million or 1 in 1 million, now the pH has shifted. And that's how we describe the concentration of hydronium or H+. We're going to use this more often. We're not going to use hydronium all that much. But in any case, as this rises, the frequency of these goes up. And so in pure water, on average, 1 in 10 million is like in this state. Okay. But as we bring other conditions in, that may influence how much we have of this. If we shift this, we begin to shift pH. Okay. If we say 1 in 10 million, that's equivalent to saying 1 times 10 to the minus 7th. And if we apply the pH logarithmic formula, that means a pH of 7. It's the inverse log of this number. Okay, so that's why it's a positive 7 as opposed to a negative 7. If you can start to get that, I know that this will throw people and sometimes people will try to push it away. Let me explain why pH becomes so unbelievably important and is one of the major things that we monitor in patients in intensive care or in their blood chemistry. So eventually we're going to talk about biomolecules, but I want to show you how pH is so important right now. And we're going to use proteins as an example of this. So proteins are made up of amino acids, amine groups with carboxylic acids and lots of side chains and so forth. But let me show you something. If you look here, we have a double bond and we have a very electronegative oxygen, and we're going to have nitrogens, and we're going to have these side chains. These single bonds here, these single covalent bonds can spin, whereas these double bonds don't. And so this combination of being able to spin and not spin, coupled with the ability to form hydrogen bonds, means that we can begin to assemble these amino acids into things like helices or sheets, which we call secondary structure. And again, we'll get to that a little bit later when we get to chapter 5, I believe. And then we can fold those helices into what we call tertiary, and then we can take subunits and assemble them non-covalently into what we call quaternary structure. All of these things, secondary structure, tertiary, and quaternary, all depend on hydrogen bonds. So if we have too few protons, too few hydronium ions, the pH will begin to rise, and we call that medical condition once you get above around 7.42 or 7.45, we call that alkalosis. If we have a pH below 7.4, somewhere below 7.35, where we have too many hydronium ions, too many protons, that's acidosis. Why is that so important? Why do we see these signs out here? Because those protons interfere with the hydrogen bonding in proteins especially. That hydrogen bonding interferes with the folding of the proteins, interferes with the structure, which interferes with the function. So even small changes, even even a relatively small change in our internal environment can lead to very, very severe outcomes in patients. If someone is hyperventilating, have you ever laughed so hard that you get hiccups? That's a great feeling, right? You get those hiccups. It's kind of funny. You know, you want to laugh because you're hiccuping, you're laughing so hard. Well, the hiccup is there to actually prevent alkalosis. Your body senses the hyperventilation, it senses a change in the blood chemistry and sends a signal down to that diaphragm to start creating spasms, which we call hiccups. It's there to save your life. So back to our picture that I showed you before with these stiff double bonds and these rotating single bonds with hydrogens, we can form these secondary, tertiary, and quaternary structures, especially if we have just the right amount of, I'm putting H+ in here, hydronium ions, what have you. If we change that, if we have too many, now we start to break up, and you can actually see the hydrogen bonds here in these dashed lines. Those little dashed lines, those little dots, those are hydrogen bonds stabilizing this, this helical structure. This is called an alpha helix. If we have too many, or if we have too few, too many or too few will alter the hydrogen bonding profile and alter the structure, secondary, tertiary, and at quaternary levels. And remember, structure is function, back to our scalable concept. We change the structure of a protein, we change its enzymatic abilities or its structural capabilities. Change those, we change a lot. And so this slide is a rehash. I won't say it again, but I'm leaving it in here because of the importance of understanding these concepts. I've explained it to you. I've shown you some pictures, that 1 in 10 to the minus 7, 1 per 10 million, and so forth. So take a look at this. This is of critical importance. Life or death importance. So important that your body actually has defenses. I like to say that carbon dioxide is as important to life as oxygen. If you have too much or too little carbon dioxide, you can't regulate pH inside your bloodstream. You can't carry the gases properly. And so pH is of major, major importance, especially if you're going to go into healthcare. And that's why we spend so much time on acids and bases, acids in our foods, acids in the drugs. We can actually control. And so I want to finish our talk today with just a little bit about the idea of that increase H plus lowers pH. We're going up here, but we're going to 0 going up. So the more H plus we have, the higher the H plus concentration, the lower the pH. The lower the H plus concentration, the higher the pH. Okay. Now, there's also OH out there, and what happens is OH and H plus will react. We have that equilibrium reaction, H plus plus OH minus equals H2O. Some of that's going to happen. So if we add OH, we're going to reduce the concentration of H, and the pH will begin to rise. If we add H plus, it will react with whatever OH is out there, and more H will be around, and so the pH will fall. I'd love to flip this upside down so that we'd actually do it, the other way, but I want you to note the scale here, that we're going lower pH as we go up here. And this is because we were showing the concentration of protons of H plus on the vertical axis. Very low protons, very high protons. Very low pH, very high pH. So don't get those flipped. Back to the idea, and here's the formula. It's the negative log. So if we have 1 unit change in pH, that means a 10-fold jump. So if we go from pH 7 to pH 6, we have 10-fold more protons. If we go from pH 7 to pH 5, it's 100-fold. It's like the Richter scale in earthquakes. It's logarithmic. We do negative. That's because it's 10 to the minus 7th concentration, 1 in 10 million. So we put that minus there just to make it a positive number. Hope that doesn't confuse you. In either case, that's where this goes clinically. And why we learn all this chemistry is because it has life or death importance. Just like the other emergent properties in water, life or death importance. So I'm going to wrap it up today. Thank you for your time, and I will see you when we move into Chapter 4. Thanks again. Bye.